Okay, I know it's a couple minutes early. I'd like to get the room quieted down because I know for those of you who have the OCAD exam, you'd love to get it out of here on time so you can get a bite to eat or something like that. So just a couple quick announcements. You are getting your exams back in discussion. You already got it today if you had discussion today and tomorrow. So means are listed on it. There are two versions of the exam, an A and a B. If you email me and say, oh, on question 23, I thought the answer should be this for various reasons, could you please specify if you have version A or B? So I'm not trying to figure out which one you have. The question order is different. The answers orders are different, things like that. So it's going to be very helpful for me if you specify which exam you have if you think there's an issue with one of the questions, okay? So just a few quick things. For the lab this week, we'll be taking and cleaning up the PCR reaction. So you did the PCR reaction last week. How many primers did you have in a given reaction? 2, a forward and reverse paired for a specific locus. So whether that was the 16S or the RPOB locus, you had 2 primers, forward and primer, that flanked it. So when we get a product, the target will be flanked by the primers because the primers determine where the reaction starts because that's where the 3' hydroxyl group is. And remember, it base pairs to a specific point on the template. Once we clean up the PCR, we'll run it out on a gel to do electrophoresis. It's going to have a positive voltage. The DNA is negatively charged. There's a gel that has a matrix in it. The DNA will be separated based upon size. Since the DNA is negative, it will move to the positive pole. Smaller pieces migrate faster. The gel allows us to do 2 things. One is to make sure we have a band. So it makes sure that once we run that gel and we loaded a specific well, we have a couple dyes in there that allow us to determine how the DNA is migrating. So we don't follow the direction of the DNA migration directly. Instead, we do it indirectly by having some dyes in there when you make your sample and load it. And what we'll see is, hopefully, you'll have a product that corresponds to the size that's predicted. And the way we'll know this is we'll have a lane with standards. So we will see specific bands appear in this standard. And from that standard, we can say the band is equivalent to that in size. Is that clear? Question. So in your PCR reaction, ideally, there should be just one band because you amplify just one genetic locus. If indeed there are 2 bands, that would indicate that either you have 2 copies of that genetic locus in that bacterium, which would be unusual, or you had contamination. Maybe you had 2 different species in there, okay? Does that make sense? So ideally, you should only have one band. So this band will allow us to determine the size. So we'll know, yeah, that's equivalent to the size we should have predicted. And furthermore, a second aspect of this, it will allow us to determine the concentration. Because dependent upon the amount of DNA, let's say nanograms, that DNA will bind to this molecule in the gel called aethylene bromide. So the more DNA you have, the more aethylene bromide binds, the more it fluoresces. So there's a direct relationship to the amount of fluorescence and the amount of DNA. So we can use this to quantify it. And that's important because when we set up our sequencing reaction, we need to have a specific amount of DNA in there, okay? So I'll show you how that works. But those are the 2 reasons for the gel. We determine that we have a band of about the right size, so we have a PCR product, and how much is in that band so we can set up the sequencing reaction. So you'll set up the sequencing reaction, we'll send it out for sequencing, and then complementation 2 is very quick. You'll do the replica plating. So as a premonition of what we're doing, the yeast strains were plated, you split them out on a YPD plate that had histidine. So everything should go right. Even though it can't make histidine, everything should grow because it's in the medium. And then we need to somehow transfer those yeast cells from that YPD plate to another plate that does not have histidine. So the plate that does not have histidine is the SD plate, and that's where we're going to be selecting for the ability to make histidine. Okay, is that clear? So if they can grow, that means they can make the histidine. If they don't grow, they can't make it. And then we're going to be doing fruit fly genetics, and I really want to spend the majority of the time on the fruit fly stuff and going over how you do genetic notation. We have a lot to do, but I think we can get there, okay? So anyway, as I mentioned with PCR cleanup, we're going to put some DNA on a spin column. There's some specific directions for this. And what happens on this is there'll be a column like this, has an opening, and there's a membrane in here. And there's basically a material in there, and it has DNA binding affinity. So under high salt conditions, so if I have some DNA and it's in high salt, and I take that DNA solution, which we hope has DNA in it because that's our PCR reaction. So ideally there's DNA in there. If it's a high salt solution, in terms of concentration, when that solution goes past the membrane, the DNA will be basically bound to the membrane. Now the DNA is there under high salt. So remember, high salt, high affinity. And then what we'll do is we'll do a wash step, which is basically to remove non-specific binding. It's kind of like a rinse cycle in your washing machine. It helps you remove residual detergent. In this case, we'll do a wash, basically. You help remove non-specific binding, things that may bound to it. Then we'll do a final step, which is the elution. And the elution is low salt. Because we've changed the environment now, we've added a low salt solution to this. As that low salt solution goes past the membrane, this becomes a low salt solution there. We lose the affinity to bind the DNA, and then the DNA will come off of that membrane and will be released into the solution there. So you're eluting it into this container there. Any questions about that? So it's a series of steps. You bind it, you wash it, you elute it. And now that once we have this, that's what you'll then use to set up a tube with loading dye. Loading, because it allows us to put that sample into this well. As a side view, this is what the gel would look like side view. But in fact, there's a little well here. And if you look from above, there are a series of wells like this. And we have a specific loading system you'll follow. It goes on and on. So you'll load it. The sample will sink to the bottom of that well. It has a couple dyes in it. And unfortunately, I don't have orange dye or blue dye. But what happens is, as you apply this electric field, we'll get partitioning along this gel matrix. So the dyes will be partitioned upon their size, and the DNA will be partitioned upon their size. So what we can see from above is with time, the dyes start to separate, and the DNA is there as well. We just don't see it at that time because we don't have enough UV light present. But we can still visualize the dye because they react in normal, just room light. Any questions about that? So we can follow the migration by following the migration of the dyes, and that should then, so if the dyes have a migration of 1,000 base pairs and 10,000, if our expected product is 2,000, it should be within that range. Does that make sense? So let me just show you some images of this. And as you know, Lab Exam 1 is coming up on the 20th, and it is 7:40 to 9:20. That's 100 minutes. And I apologize for the lateness of that exam. We actually had a much earlier time, but to allow chemistry to get an exam that night, we had to move our exam back. The same is true for our exam on May 1st, which is 8 to 10. Again, so chemistry could get exams. So PCR cleanup, not only are we binding the DNA here, but we're also removing the primers. Because when you did that PCR, you had forward and reverse primers. You did not use all of them up. They're still in that solution. But when we send this out for sequencing, we need to have a specific amount of only one type of primer. So when we do the forward reaction for sequencing, we'll have only the forward primer. But since the PCR reaction had both the forward and reverse primers, One aspect of this is the primers are small, so they don't bind to this membrane. So we get rid of the background primers, so that when we finally get to the solution, the primer should be gone, so that we can set up the right concentration for sequence reaction, and then add a specific primer and only one primer present. For sequencing, only one primer type, a forward or reverse. All right, we've gone over binding affinities, the final solution with water at low salt. The two samples are the two PCR reactions. Any questions about this? Just let's see how this works. There's our PCR product. We bind in high salt. There's that membrane. This just shows you it's like a gel matrix. So under high salt, high affinity, the DNA is bound to it. That is the PCR product if it's present. The primers are so small, they do not bind. So the primers just go right through, but then we'll do a wash step again to remove non-specific binding. Then the final step is the elution. Low affinity, the DNA will be in this tube now, ideally. You have no idea if there's even a product present. You certainly have no idea the concentration of that product. So that's the purpose of the gel. This shows you the gel. Here's that loading dye. It has the two dyes in it. It's also got some solution that makes it dense, so it sinks to the bottom of that well. Here's the positive pole. The DNA is negatively charged. So are the dyes, right? And they migrate to the positive pole, and with time, they separate more and more. Just like with the chromatography that you did with the green, the chlorophyll extract, right, the pigment extract. When you first put it on there, all those pigments are in one band. But as they move up, they partition between the two phases, okay? So this isn't actually partition between the two phases, but we'll get that separation. We've talked about this a little bit with the sequencing reactions. When you looked at the sequence, the autoradiograms last week, the photocopies of them, you saw exquisite resolution. So resolution refers to how small can these DNA pieces be and still know that they're different sizes. So with the acrylamide gel that we used in sequencing, we knew this was one nucleotide long 2, 3, 4, 5, 6, 7, 8. Exquisite ability to resolve it. The gel matrix there was acrylamide, polyacrylamide. Okay? So this has exquisite ability to resolve. And we see the bands, that as we visualize them, we can see they're present because these are radioactive, and we put them on an X-ray film. In this case, we're going to use an agarose gel. We get the separation, but the separation is not nearly as fine detailed as with the polyacrylamide. It's a much looser matrix. Okay? It's kind of like when you make Jell-O shots, all right? I gotta speak in student language. Anyway, with Jell-O shots, you add extra gelatin to make it nice and rigid, right? If you don't add enough gelatin, it's basically like water, right? So you can add more and more gelatin to make it more and more rigid. So likewise, you can change the concentration of acrylamide to make this a finer mesh, better to resolve, and likewise here. But again, with an agarose gel, we don't get great resolution even if we really use a really concentrated form of it. So if you notice here, these are about 280 apart. We can sort of see that, but if they were within 25 of each other, we'd probably never see that difference. Those two bands would blend. We would not have the ability to see them as two distinct sizes. Here we can, here we can't. And when we have those standards on there, we could generate a standard curve like this if we wanted to, because when we have the standards that we load, we know the sizes of them ahead of time, and we know the amount of DNA that should be in each band. So we could figure out the distance it's migrated, and say, okay, we know the distance it migrated, we know the size, we know the distance, and you plot this and come up with a standard curve like this. And that is, the bigger the piece, the less distance it should migrate. The smaller the size of DNA, the farther it should migrate. You can come up with a standard curve like this, and then you can take an unknown band in terms of its size, but you can measure the distance it migrated from the gel, just with a ruler. So since you know this, you can determine that. You've used standard curves before, for example, in the first experiment when you tried to figure out the concentration of the dye. It was pre-made for you. You also used it with the enzyme lab to figure out how much amylase activity was in the saliva. Okay? You actually won't have to make a standard curve this time because you'll see that your band is ideally between a couple of these and say, yeah, it's there, it's about 1200, okay? But if we had multiple bands there, you ideally would make a standard curve. This just shows you the DNA here, and you see it fluorescent in the presence of a UV light, and there's a thinning bromide in this gel. So the thinning bromide is a molecule, and it basically intercalates between the base pairs. So remember you have that double strand like this. If you want to think of it like a ladder like this, those bases form planar bonds like this in the middle, planar structures. So the thinning bromide goes between those bases, those planes. So the longer the piece of DNA, the more thinning bromide can bind. All right? And that's what this just illustrates. A longer fragment would be able to bind more DNA. So if I had just 1,000 pieces like this, 1,000 like that, 1,000 like that, 1,000 like that, this band of 1,000 that's longer would fluoresce more than this band of 1,000, this would fluoresce more than this band of 1,000, this would fluoresce more than this band of 1,000. Because the total number of base pairs is greater in this band than that. But I also want you to realize that if I had on that gel something like this, if I have a band here, let's say 500 molecules, and those are 2,000 base pairs long, which direction would and the migration is this way, which direction would I find the base pair, the band that was 1,000 base pairs long would be down or up relative to this? Smaller, so should it migrate faster or slower? Faster, so it should be down. Relative. And if I had 1,000 molecules that were 1,000 base pairs long, which band would fluoresce more, A or B, or would they be the same? The same, because the total amount of DNA is the same. 500 times 2,000 base pairs means there's a total of 100,000, sorry, 1 million base pairs, and here there's a total of 1 million base pairs. Since it's the overall same number of base pairs, these two would fluoresce to the same amount. Does that make sense? So on the lab exam, I could give you a pattern like this, and I could say to you which band should fluoresce more if I give you this information. Does that make sense? So it's, again, remember, fluorescence is dependent on the total amount of DNA, which is going to be dependent upon the number of molecules and the length of those molecules. All right? Excellent. This is just the gel you'll be using. They're precast. They're very easy to use. This is the direction there. There's the wells. They'll migrate to the positive pole. It's pretty quick. Again, it allows us to see, do we have product? It allows us to see the size. It allows us to determine concentration. Now, let me just go over how it allows you to determine concentration. Just bear with me for a minute or two. So here's the laddered. So we see this is about 500 base pairs with 0.5 kb. This is a band that's about 1,000. This is a band that's 1.5. This is a band about 2.0, etc. These are in kilobases. If you notice, this fluoresces the most. Now, if you notice here to the right, we give a mass of this. So there's 200 nanograms in this band. So if our unknown had the same amount of fluorescence, we would know in that band of the unknown there was 200 nanograms. If the band of unknown matches this fluorescence, we would say it's about 75. If it matches the fluorescence of this, about 70, or this, about 50. Okay? And we're just kind of getting general ballpark. Okay? We're not going to say, oh, it's 53 versus 55. Call it 50. Okay? All right? Now, when you set this up, you took 5 microliters of your elution. So when you set up your loading dye, there was 5 microliters in this. So when you figure out the concentration, you have to remember, if you want to get it per microliter, that you have to take this divided by 5. Does that make sense? Because you used 5 microliters of this elution to set up your reaction that you loaded on the gel. Therefore, this band is a result of 5 microliters. So whatever this concentration is, you have to divide by 5 to get the concentration per microliter of this elution. Okay? Any questions? Yes? Okay, so it depends. Okay? So let's say this is what it should have been. So the question is asking, what does it mean if I see 2 bands? Okay? So first off, in order to tell that they're 2 bands, they would have to be 2 different sizes. Otherwise, we wouldn't know. If there's lots of this, I would say that's a lot of product of that one size. So to see 2 bands means they're 2 different sizes, which tells us when we did the PCR reaction, we had one locus like this, flanked by the primers, and that yielded, let's say, band A. And somewhere, I had, maybe it was another piece of DNA, because it was another species, and I had this, where the primers annealed, and that gave me band B. The other possibility is that if I somehow had a gene duplication of this, somehow I duplicated gene A and had it here, but somehow the DNA had some insertions, so basically I'm expanding this, so this becomes bigger, that moves my primers further apart, which might explain why I have this bigger piece, okay? Now, this is probably very unlikely. It would most likely be that you had 2 different species in there when you started it, which is why we were so careful to have it streak and streak and streak, okay? Now, that having been said, we've never really seen 2 bands. The other possibility is, when I said there's this contamination like this, we used a polymerase to do this reaction, right? Now, that polymerase came from some organism. So it's maybe possible that the colony wasn't actually 2 different species in the colony, but this may have been a contamination in the polymerase prep. That is, when we purify the polymerase from the bacteria, that polymerase has a bacterial genome, and we think we've purified the polymerase sufficiently, but we may not have. And in fact, there may have been contamination in there from that original starting bacterium that the polymerase came from. And this is a contamination that is real. It does happen. But ideally, you do a control to look for that, okay? Because you would still get this band in the absence of any template, right? Because the template was present in your polymerase prep. Okay, does that make sense? So in fact, there are controls you can do to eliminate that, all right? A good question. When we do science, we have to do lots of controls to make sure that what we get results is what's expected. All right, so just to calculate the volume, you want to have 100 nanograms of DNA in your reaction. So if there was 20 nanograms per microliter, then you need 5. If there was 10, then you would need 10. The bottom line, though, is the most you can ever add is 12, because the final volume that you want is 12 microliters, because we will add 1 microliter of primer. The GSI or UGSI will add that primer. So what happens is when we send this out to the sequencing facility, they don't know what primers we used. So when we send it out to them, we'll give them a tube that has 13 microliters. 1 microliter of that will have been the primer. Forward if it was a forward sequencing reaction, 1 microliter of the reverse if it was a reverse reaction. And so you have 12 microliters that you can play with for your DNA. If your DNA is, concentrated enough that you only need 6 microliters, then you make up the volume of water. If we have some pure water there purified. Is that clear? And if you remember last time, again, remember the sequence and the way it works. When we have the dideoxy present in the synthesis, it stops the reaction because there's no 3' hydroxyl group to attack. And as a result, that's what stops it. Now, when we send this out for sequencing, they'll be using the 4 colors here, so we will do fluorescent sequencing there. And again, the primer gives rise to the 3' end. It dictates where we start the sequencing reaction at. And remember, there's a subset, a ratio of dideoxys to deoxys. And in this case, all 4 in one tube. Okay? So we have all 4 types of deoxys in that tube and all 4 types of dideoxys. But whenever this gets incorporated, it stops, it gives us green. Whenever this gets incorporated, it would stop, give us red. Whenever this gets incorporated, it would stop, give us black. These aren't the real colors, by the way. And this would stop and give us blue, okay? And then when we get the data like this, we'll see the different colors as it migrates, and we get a chromatogram trace like this. And that's what you'll get back is actually a trace like this with numbers on it, so you can keep track that this was the 100th position, 110th, etc. Any questions about that? And all that's done for you, and that's basically it's tied to a computer, and they just give you the chromatogram. All right, so questions on the lab exam in terms of how can you apply this concept you've learned to things. I can have something like this, where I just give you this and say, what's the sequence of the strand that was made? Or I can say, what was the strand of the template? And remember directionality, that is the 5' versus 3' end, okay? And this represents the 5' end of the growing strand, because it grows 5' to 3'. So number 1 is here. The first nucleotide incorporated, the second would be here, third, etc. So we're going from 5' to 3'. So the smallest would represent the 5' end we read up. That's of the strand made, and of course the template is anti-parallel and complementary. Okay? But realize I could sometimes add 2 dideoxys in a tube and ask you what would it look like. Or I could say, whoops, you forgot a dideoxy, what would it look like? So there are lots of questions I can ask about this where you apply the general concept. And when you look at the exam reader, you have 5 sample exams. You're going to see various permutations of this, okay? So, by the way, next week is bioinformatics. I'm going to ask you to watch the lab lecture. I'm going to give you a link to an old lab